Here. All right. Welcome everyone to this Renegade Research Roundtable. If this is your first time with us, thank you for joining us. I'm Isabel, CEO of our organization. We are a patient and caregiver-led, decentralized 501c3 nonprofit organization focusing on complex, multi-system chronic conditions such as ME/CFS and long COVID. Within this, we have several areas of focus: research that is agile and patient-centered, coaching and telehealth to provide care support, and education to increase the number of clinicians treating us. Our work is made possible by a wonderful team of volunteers who move the wheels of our organization. Most of us have a personal interest in advancing research and care of these conditions. To stay in touch with us and hear about upcoming events, please subscribe to our newsletter and follow us on social media. We will be posting those links for you. As a nonprofit, our work also depends on donations of all sizes. You can help us fund our work by donating to our funding campaigns and also by sharing our campaigns with others who can donate. To raise funds, we also have some affiliate discounts by partnering with companies whose products and services we have used and like. This does not constitute advice on using these products and services, and as always, discuss with your healthcare team. If you do use them, please consider using our discount codes to help fund our nonprofit work. And now I will pass it up to Tess to introduce today's roundtable presenter.
Hi, I'm Tess. I'm Chief Science Officer at Renegade Research, and I'm really excited to have Nick here today. We've been following their work for a long time, and we're going to be collaborating with Amatica for the upcoming project we have, which you might have seen on X. So yeah, I will introduce Nick, and just excited to have you here. Nick is the CEO and co-founder of Amatica Health. He's a neuroscientist and bioengineer specializing in genetic and cell engineering, synthetic biology, neurotherapeutics, and data science. So go ahead and start sharing your slides and we'll get started here.
Thank you so much, Tess. As always, a great pleasure to be invited here and to share this space together. I've also, as you know, and everyone I hope knows, have been a big fan of Renegade Research since the very beginning. I'm very excited about our upcoming work. Thank you everyone for joining today. I think my slides should be up.
Yep, we can see them.
Great, thank you so much. So thank you everyone for joining. Before we go into today's presentation, for anyone who is new to Amatica, I'd like to give a short intro into what we do. So we basically are originally a patient-founded research company in this space. We originated from networks of patients who wanted kind of self-actualization and wanted to understand their own disease and biology. And it's grown into a patient-driven research model where basically we offer really deep tests directly to people, individuals, that can give them kind of a unique level of insight into their own health and biology. And in turn, those tests add data to our database, which drives our research. The research approach is mostly centered around protein and RNA testing. The subject of today's presentation is all going to be findings from our RNA cohorts. So for this, we do RNA sequencing from whole blood. And so for that, patients buy an RNA sequencing test. We collect their whole blood sample, extract the RNA from that sample, and then collect really rich patient phenotyping data on their symptoms, disease history, disease manifestation, and then we integrate that in our database to build an open-ended, linked biological and patient-reported data set. And in terms of our research goals, fundamentally, this graphic here represents, I think, the kind of core of what we want to do. So we believe that these complex neuroimmune diseases like long COVID and ME/CFS, among others, we believe that they are characterized by a really heterogeneous patient population. So I think the best evidence for this is that, and I think we, as patients, I'm a patient myself, I think we've all experienced this, what one of my co-founders, Jack, calls patient roulette, or treatment roulette rather, where we will hear reports of a treatment helping one patient, and then 20 or 30 other patients might try that treatment and have no response, and another patient might have a significant bad outcome where they have a baseline drop or are permanently worsened by that intervention. And so that suggests that if you have this extreme diversity of manifestations, different disease profiles, different symptoms, as well as clinical outcomes, it suggests that there must be underlying biology that drives those differences in outcomes. So, our belief is that the patient landscape is defined by many different biological subsets within it. Our research goal is basically to stratify that patient population, meaning separate it into those biological subgroups, and then study those subgroups, identify signatures within those subgroups that can separate them, and then study and then use those signatures as a way to predict treatment matching. So, matching people with a specific profile to a specific drug, as well as researching disease mechanisms themselves and trying to identify novel drug targets and treatment approaches. So as I mentioned, today's presentation is going to focus on our RNA sequencing dataset. So this dataset today is now contains around 234 samples. That includes controls. So it's about 149. So I think it's around 165 samples, where 149 of those have completed deep phenotyping, so the really detailed questionnaires that we accompany our tests with. And those samples split like this. So the grade that says not surveyed is actually also the control population. And then we have a split where we have MECFS in blue in the middle, we have long COVID and MECFS together, and then in turquoise here we have long COVID in isolation, and then other diagnoses. So that breaks down like this. So we have around 60 combined long COVID and MECFS patients, around 59 patients who list only MECFS without long COVID, 19 who list only long COVID, and then 11 who list other things like vaccine injury or some other adjacent diseases. In terms of onset, I think in our research, we believe that we don't believe in the definitions, right? So we think that the definitions are, they're not unhelpful, but they don't capture as much information as we want. So we try to look at that at various different levels. So firstly, if we look at the triggers that led to these disease manifestations, you can see the distribution here. So this was a multiple selection, and you have 51% of people citing a viral infection, 49% citing COVID, but then many others that I think that patients can all really relate to. So for example, psychological stress, 28% of people cited psychological stress as a contributing factor to the onset of their disease, vaccination, 18%, and then even physical trauma, 9%, which is really significantly high. I think many of us have probably heard about surgeries or other physical traumas being a contributing factor to the onset of MECFS. Equally, 55% of patients said that the onset of their disease was delayed, which I think is, one of these, interesting things about these diseases that really speak to their complexity. My onset was 5 months after my acute COVID infection, and other contributing factors kind of piled on in those 5 months. Looking closer at post-exertional malaise, we're really excited to be working with Renegade Research to do some really important research on post-exertional malaise. Recently, I think a huge percentage of patients cite post-exertional malaise as the single problem, not for everyone, right, but for a very significant portion of patients. They could live with every other symptom except post-exertional malaise. And so firstly, we looked at this in a few different ways. So just from a very high level, how did people rate the severity of their own post-exertional malaise? You can see that over 50% of patients listed it as severe or very severe. And then PM onset delay, I think this again is a really interesting feature that again speaks to the complexity of this symptom, is the fact that the majority of patients experience a delay between trigger and symptom onset. And so that delay, has a distribution like this, and you can see that over 50% of patients have a delay of more than 6 hours before onset. That really suggests, some kind of second order effects, whether it's gene expression changes or cell system state changes, things that we need to understand. But it's a really prominent feature of PEM as a symptom. Recovery time can span hours, days, weeks, months, and years. This figure, I think, should include years as well. And then overall, going back to the disease as a whole, we also look at how it impacts people's lives. And, I personally find that looking through this data is always really important. I think it's like, for me, both as a patient and as a researcher in this space, I find it really important to look at the data on just how, how severely affected patients are in this space. So if you look at functional status in our cohort, so in this, 149 patient cohort, 63% of the patients included are either housebound or bedbound. So they're either at best housebound, 27%, or either mostly bedbound or completely bedbound. This is really significant because, for our research, there are many limitations to our research, and we face, many unique challenges because of the way we approach things. But we also have some advantages which, make us, give us a unique ability to study these diseases. And having more than half of our patients housebound or bedbound, we think is really important. Many studies that we see in the public literature are only accessible to patients who can go to a location to give a blood sample. Many patients cannot do that. So, it's a real asset to our data set that we have such a significant portion who are housebound and bedbound. In terms of impact on work and education, 53% of our patients are totally unable to work or pursue education, and 80% are unable or either unable or significantly impaired from work or education. In terms of functional capacity, I always find these really shocking to look at. So 36% of our patients are unable to walk more than 500 steps a day. I'm currently in that category. 50% of patients are unable to walk more than 1,000 steps a day. And for all of the patients who are living in this situation, and for anyone who doesn't have perspective on this, 500 steps a day is not enough for a normal day of moving around within an apartment, a small apartment or house. So this is really where you have to be planning trips across rooms and trips to bathrooms. So, it's really quite shocking just what a significant proportion of patients are living in that way. Talking capacity, I also find really shocking. So 13% of our patients are unable to talk for more than 15 minutes per day, and 46% of our patients are unable to talk for more than 1 hour per day. In terms of impact on daily life, we collected data on what daily activities people are able to complete without assistance. So, if you look at this from the top down, 89% are able to eat without assistance, but that means that 11% are not able to eat without assistance. And then when you get down to, light housekeeping, walking, transport, the majority of patients are unable to do those things without support. In terms of the impact of ME/CFS on people's lives and well-being, 99% of people say that it's impacted their physical health, 89% of people say it's impacted their work, and 85% of people say it's impacted their social lives. Finally, there are many other areas we could go into detail on, but I think just to finish off this section, in terms of cognitive difficulties, it's cognitive manifestations of this disease represent, I think, the biggest cluster of different symptoms, right? There are so many complex neurological symptoms, and some patients have literally dozens of cognitive symptoms. From our cohort, 86% report problems of concentration, word finding 71%, short-term memory 70% have issues. So looking at integrating this data, we've come up with some insights into the perspectives from the cohort as a whole. So in terms of symptom frequency, in terms of symptom severity, averaging across all patients, ordering symptoms by severity, interestingly, in our data set, sleep emerges as the symptom that people self-report as the most severe, followed by cognitive dysfunction, autonomic dysfunction, neurological issues, pain, muscular, etc. This doesn't include PEM, by the way, because we have that as a separate category of its own. We also looked at symptom co-occurrence. So we looked at across the major symptoms areas that we collected, where did the severity of each symptom co-occur with other symptoms? And this is a summary of the associations between different symptom severities. And really, some of these areas, we need to look into in more detail and build a clearer picture of. But just as a couple of headline things for interest, one of the most interesting associations was that the severity of muscular symptoms correlated very strongly with the severity of neurological symptoms. And that I think is really interesting because in the list of possible explanations for the muscular manifestations in long COVID ME/CFS, I think neuromuscular junctions are definitely a strong candidate. So something that we will return to and look into more detail later. We also looked at symptoms versus disability. So we took the disability scores. We created a disability score from how people's overall severity. So that was bedbound to being able to walk normally, and we correlated that against different symptoms to see which symptoms were associated with the highest level of disability. Unsurprisingly, PM has the strongest, extremely strong Spearman coefficient of 0.8 with the level of disability. And then looking down from there, it's also really interesting. So neurological symptoms were the symptom cluster that tracked second strongest against the degree of disability. I think that's really interesting. I mean, again, it points to the fact that, our patients cite sleep as the most severe of their symptoms, except for PEM, and neurological symptoms track strongest with disability. And if we look at treatment responses, I think there's a lot of patients who are finding that neuromodulatory drugs are, moving the needle, most of them. So again, it really points to this strong neurological kind of center in this disease area. We also looked at burden versus severity, so the relationship between how many symptoms people had within a cluster versus the severity of those symptoms. And what I think is most striking here is that cognitive dysfunction is the combination of the highest burden, so the number of symptoms people had was the highest of any area, and the severity was as high as any other area, including sleep and neurological symptoms in parallel. This, I think, is really fascinating. So, I think everyone who follows our mission knows that a major part of our research is that we want to collect treatment outcomes, collect treatment responses, and then try to identify signatures that can predict treatment responses. So, in our cohort, we collect detailed data on people's response to different drugs and different treatments. And this distribution here shows for the treatments that we're collecting data on in our onboarding questionnaire that most of our patients have completed. This plots the relationship between the responses. So in the red side of the bars represent the percentage of patients who worsened, and the greens represent the percentage of patients who improved. And then the number in the red bars is the number of patients who have tried those drugs, right? So in the questionnaire, patients asked, have you tried this drug? And then if you have, what was the outcome? So this is a really interesting insight because it shows here, and again, there are limitations here. This is just preliminary data, and this is not, independently validated. So please don't use it as a reason for how you make your decisions. But it's really interesting insight into the kind of risk, benefit, and jeopardy of different drugs, right? So if you look at the bottom of this figure, COVID vaccines is a kind of confounded category. So just that one have an asterisk next to it because many people's disease was triggered by COVID vaccines. So this one is, maybe a little confounded by that. But, for example, rapamycin, 14% of people reported improving, 41% reported worsening. It's only 22 patients. Now, it's a small sample size, but it is certainly interesting. From 22 patients, 8 got worse. And I don't know how many people have seen the rapamycin preprint, which has just been released recently, showing really detailed patient questionnaires at 30, 60, and 90 days post rapamycin, and that study seemed to have quite promising results across several domains. And I think that our data certainly conflicts with that at this small and very narrow scale. But I also think it's a reason why we should be putting strong emphasis on looking at worsening and wanting to understand worsening. I would say that internally for us as a company, patients worsening from treatments is maybe more important than patients improving. If we could eliminate patients worsening, then patients would be more confident to try new drugs. I mean, that would be, I think, the first major step, the biggest possible priority. At the other end of the spectrum, I think the drug that had the highest percentage of people reporting improvement was benzodiazepines, and those, it's not surprising to see. It's always it's a tricky one because I think that rightfully so, many people are afraid of dependence and abuse of those drugs. I certainly am myself. But it's interesting that they come up so highly and from a pretty large number of people from our data. We also looked at treatment responses by disease features. So we looked at so this distribution firstly shows the relationship between disability and PEM recovery. So it just shows that people who had longer PEM recovery also report higher levels of disability. And then we also asked the question, do more disabled patients respond less to drugs? So this shows the percentage of treatments that patients had tried and reported improvement against the level of disability. So ambulatory means that people can walk normally. Housebound obviously is people who are stuck within the house, and bedbound, bedridden is mostly bedbound or fully bedbound. And maybe unsurprisingly and slightly sadly, the patients who are more disabled have a lower reported treatment response in this cohort. So again, underlining the need for research into those more severe patients and assistance in them in choosing the right treatments. We also plotted a relationship of treatment risk benefit. So from our data, we plotted the space of worsened and the degree of worsening versus the degree of improvement. So drugs that are in this bottom right quadrant are the ones where the highest percentage improved and the lowest percentage were worsened. We will be releasing this preliminary data after this presentation as well. So that is just a summary, a kind of high-level overview of what we see in the type of patients in our database, and I thought it was an interesting starting point for this, for the first look into some of our research into our RNA sequencing cohort. So I'm going to focus on 2 sections for the rest of this. Most of the time today is actually going to be spent on this section, which is exploratory classification analysis. So this is AUC research. This is the beginning of research towards signatures that can separate disease groups. So, the research towards candidate diagnostics, for example. And then after the section, I'm going to cover just a little bit of first insights into some of our, broader research into our RNA sequencing cohort. So this exploratory classification work, it's been a real priority for us because on our mission, given that our kind of key North Star is eventually to be able to stratify the patient population into different biological subgroups, a really important proof of concept of that work is, can we separate the disease as a whole from controls? And at the scale we're at now, so this 149 sample cohort, that isn't enough samples to separate, many small subgroups yet, but it is enough samples to be doing cohort level separation. So as a proof of concept of how we plan to use this data set when we get to 500, 1,000 or 2,000 samples plus, this serves as a real validation of our model. So for this work, just important for me to say that this is work in progress. This is all preliminary and exploratory. We are working on this currently. We're still developing this work. We've got another 52 samples joining this cohort soon, and we're really excited to see the expanded performance. And we're preparing this for publication, and we will be putting out a paper on this hopefully later this year. At which point we'll be really interested to get feedback from the entire community. So our current progress is we've managed to achieve an AUC of 0.893. So that means that our signatures are able to correctly classify people into either patients or controls 89.3% of the time with a p-value of 0.01. So, the point in this research is that there's no validated diagnostics in ME/CFS or long COVID. Still, no one has brought an overall diagnostic to clinical adoption. And when we started Amatica, there were teams around the world who had already published preliminary diagnostic signatures. And 3 years ago, I thought, okay, well, there's no way Amatica would ever be the company to make the overall diagnostic. Every year that passes, I have to wonder if we eventually will. We're certainly doing the research. Like I said, our real goal is not overall diagnostics, is to separate subgroups. We will see what things look like as we progress. So for the question for this, we asked, can we find objective signals from whole blood RNA sequencing to discriminate the disease groups? This is the cohort, so 244 samples, 159 patients, sorry, not 149, 159, 85 controls, age and sex matched, included in every sequencing batch. And as I mentioned, 63% housebound or more severely affected. It was whole blood collected in PAX-gene RNA tubes, rRNA and globin depleted, and then we ran it on 100 Novosec X 150 base pair paired end reads to a read depth of 60 million reads per sample. The way this research works is we're using an algorithm called GLMNet, which basically looks for, so it's an algorithm that looks for signatures that can separate groups, and it penalizes the number of data points used. So it tries to solve for a small group of signatures that can discriminate groups. The way we do this is we build a training architecture represented here. So the cohort is split at random into 5 groups, so of 20%. 80% of those samples are used for training the algorithm, and 20% are held out so the algorithm has never seen them. And then after training, the algorithm is used to assign the held out samples, and then the AUC that we present is the percentage at which those are correctly discriminated into the different groups. This is then shuffled and repeated 10 times, and then the AUC is averaged between those runs. Before starting, so the best way to approach work in this area is that you define the acceptance criteria before you start. So we're researching that, and we landed on, and we defined what we considered acceptable thresholds for all of the key features before initiation. We also ran at every stage 4 parallel models. So we ran age and sex alone as a kind of clinical flaw. We ran cell composition alone. So this is cell type deconvolution estimated in different cell types, to check that it wasn't just cell type driven. And we also ran all the technical variables alone. And then finally, we ran the RNA expression data alone as the real signal. The first run, so this is just literally the results from of running on the kind of default settings for these algorithms on our first pass. We saw that age and sex had no predictive power, cell composition had essentially no predictive power, and technical confounds also had very low predictive power, and yet RNA expression already had 88% ability to discriminate samples on our first pass using this GLMNet algorithm. We then went to work trying to refine and tune and improve. So from there, we did a few kind of steps to try and improve the predictive power. So firstly, one of the first things that we thought would be a contributor here was potentially a noisy gene flaw. So just a bit of background here, in RNA sequencing, when the output is counts of each gene, so these are the RNA copies of each gene, and sequencing counts the number of copies. The number of copies tell you how much of a protein is being produced from that gene, and the counts have much higher stability when they're higher, right? So low counts below 1 are very noisy. Counts below 10 are also noisy. I mean, just for perspective, in counts below 10, we see a variance of, I think, roughly 70%. Below 20, it drops to kind of, I think, 30, 40%. Below 50 and below 100, it drops to 20%, and above 100, the variance is around 10%. So the signal is much more stable over 100 counts. That's really important when you look at your RNA sequencing data. So by cutting the flaw, we were able to get the AUC to increase and up to 0.886. We then tuned the model. So the model, like I said, it penalizes the number of genes used, but there you can tune how strong that penalization is and whether it optimizes for more softly associated genes or less very independently robust genes. In our case, we found that optimizing for more softly associated genes increased stability above our minimum threshold and retaining the strong AUC that we wanted to see. Combining those two approaches, so cutting the gene flaw at 100 and tuning the model, got us up to an AUC of 0.894. So as I said, that's 89.4% accuracy at discriminating samples into the correct groups. We then ran a permutation test. So this is to figure out the actual confidence of a result. So to do this, we scrambled the labels. So the label of disease and patient was shuffled for all the samples, and then we ran that 97 times. And then we ran our model with the labels correctly assigned, and our signature was able to discriminate perfectly. There was no overlap with the random permutations, and then this gives us the p-value of 0.01 for this preliminary AUC. We then looked at potential confounds. So we went into trying to understand, could any other parameters be confounding these results, be driving these results when it isn't real biology. And so we removed correlates of age and sequencing depth, and those only reduced the AUC from 0.894 to 0.893. So that was now our confound-free, stable, and robust signature. And so, as I said, this is preliminary work. It's in progress. I think that 6 months or a year from now, I hope that we have really, really robust, validated diagnostics and stratification signatures. But as a snapshot of where we are today, we've achieved that 0.893 AUC with high confidence. If you look at the breakdown for our different conditions, so combined ME/CFS and long COVID is 0.893. ME/CFS on its own is 0.903, and long COVID on its own is 0.858. Long COVID is currently a little underpowered compared to ME/CFS and combined. And in terms of sensitivity and specificity, so this is the rate of false negatives and the rate of false positives. You can see that this is not at the level of a clinically ready diagnostic, but it is still really promising progress in that direction. So the best we can do is 92% sensitivity and 78% specificity. So that would mean that if you were using this, and if this reproduced and was validated, just for understanding, this would mean that there'd be a false positive rate of 7.8% and a false negative rate of just under 30%. So, not yet quite. You'd want higher specificity, and ideally, you really want sensitivity and specificity to be over 95%. But certainly, it is a really, really good progress in this direction. going back to the acceptance criteria, so it met all of the acceptance criteria that we set. So, that met our own internal benchmark. And then in terms of where we are, so as I said, this is preliminary work in progress. It's one cohort, it's internally cross-validated. We have this no leak discipline, so it's always trained on data and then tested on data that it's never seen before. But it's not externally validated yet. And, it's at the scale of 159 patients and 85 controls. That actually is, a relatively large scale. Most of the studies in this space that have published AUC results have been on smaller cohorts, and that's really significant because it's much easier to separate smaller cohorts than larger cohorts. So this is encouraging, and our patient pool will rise to 211 in the next month. So it'll be really interesting to see, does that, do we retain performance or improve, or can we find new features? The key milestones that will come next is, obviously, eventually independent external validation. And, if we really do take this all the way, or our stratification diagnostics, then formal validation. Next, does increasing the size, hold and improve? We're preparing a paper on this, so we will be, continuing this work, and then, we'll see the performance after the next 52 samples are added, and then we will prepare a manuscript and submit with a, likely with a preprint released at the same time as submission. And then, more interesting questions for us in the future will be, can we separate patients by severity, or most interesting, by disease subgroups? And so, that disease subgroup work, I think many people who follow our work know that's a major focus for us. And I'm going to just look into a little bit of that in a minute. So that's our AUC work. And finally, I'm just going to show some of our early RNA sequencing findings from our cohorts. So as I said, we wanted to look at the AUC diagnostic work first, because it's such an important proof of concept for our model to be able to discriminate groups. But of course, in parallel, we have this incredibly rich RNA sequencing data, and we're collecting this really rich patient phenotyping. So of course, we also want to do, research into the disease as a whole, whether that's looking at altered biology, but especially looking at at distinct biological groups and subsets of our patient population. So in terms of cohort level findings, I think that we will continue this research over the next months, and probably we'll do some more seminars, maybe in 2 or 3 months, where we really go into our research, our overall research from the cohort. But I want to give a little preview of that and a kind of snapshot of what we see at a cohort level today. So firstly, in terms of differentially expressed genes, so these are genes that are significantly differentially expressed between control and disease, and that survive FDR for multiple testing, and so it's p-adjusted under 0.05. We see over 3,000 differentially expressed genes in the combined long COVID and ME/CFS cohort, and then individually in ME/CFS 2,000 and long COVID 1,500, probably a little bit underpowered for long COVID. In terms of validated pathways, so this is where, instead of looking at individual genes, we use a I think probably the best annotated pathway library called Reactome, which is a library that organizes genes into the pathways that they belong to. So these could be signaling pathways, or transcription pathways, or cell maturation pathways, and those pathways aggregate 10, 20, 50, 100, multiple hundreds of genes. And so by analyzing them, you look at not single gene expression changes, but you look at coordinated shifts across entire pathways. That will become a really important part of our research, and we're actually going to be releasing that to all of our patients soon as a feature in their platform. But those pathways can reveal, like much more robust biological insights by looking at that coordinated activity. So as a snapshot, this is what we see at the cohort level. This analysis, so this is just quite a narrow analysis. So this is looking at really small subsets of genes for some major systems of interest. And so these are small gene panels. For example, for mitochondria, it's a panel of 32 genes when in reality there are over 1,000 mitochondrial genes. So, this is a narrow snapshot, and this is averaging at a cohort level, right? So this is, taking people potentially with very different biology and averaging them together. I add those caveats because, I think we will see, much more significant differences in all of these systems in the middle. But before we looked at it more, in more detail within each system, we wanted to just get a quick approximation of any really significant changes that we see at the cohort level. And the reason I'm showing this is because with those caveats, there is a really significant shift in neutrophil genes and in B cell genes, and they're both interesting. So I thought we'd just look at them for a minute. So in terms of B cells, it was the B cell and antibody related arm that was at a cohort level was significantly down. And so these were B cell receptors, immunoglobulin genes, master B cell transcription factors. And it actually suggested a kind of an apparent reduction in the adaptive antibody compartment at a cohort level. Conversely, neutrophils had a signature being up of degranulation of emergency granular poiesis, and they had upregulation of genes involved in their maturation, activation, and pathogen responses. So it's a signature of kind of expansion, activation, and immature neutrophils, emergency granular poiesis. And really interestingly, we do cell type deconvolution as a part of this analysis. So we look at the predicted ratios of each cell type in the data set, and we use that to, we include that in all of our analysis to correct for cell type shifts that may contribute to apparent gene shifts. So it's a kind of important quality control step. The neutrophil cell compartment was actually found to be reduced in the cohort overall. So this result is especially interesting at a cohort level because we found the neutrophil compartment, the number of neutrophil cells, the predicted number of neutrophil cells was down, but the activation markers and maturation markers were significantly up. So again, like I said, this is a narrow panel of genes for each area, and these are the averaged cohort level findings, but still really interesting. It does suggest that at a cohort level, there is a neutrophil activation maturation bias, right? There may be more patients who have an increase in those features than those who don't. The B cell one, I think, is presenting, I think is as a cohort level finding, is obviously preliminary. It's certainly interesting because we know that, I think we have quite strong evidence that B cell mediated autoantibody processes may be contributing to symptoms for many patients. So when we saw these results, my first thought was, okay, we're looking at the cohort level averaged, meaning maybe there's a skew over at a cohort level towards these features. But what happens if we separate patients by their immune cell states? Do we see a spectrum, and do we see different clusters that are more complex than the overall figures suggest? And the answer is we do. So what you're seeing on the left here is clustering for all of, for these, narrow panels of genes within each of these immune cell compartments. Patients are being clustered by those genes, and you can see that there is basically an entire spectrum where there are patients who are significantly up and there are patients who are significantly down. There is some separation into broad clusters here. And, what we will do is follow up looking, system by system, and, do we see more clear groups separating out? But if you look at the table on the right, this basically shows, for each of these compartments within these clusters, what is the kind of z-score, the kind of how many standard deviations are you away from the mean? So how up or down, are these systems overall? And that reveals that there are basically two clusters where, if you take, for example, monocytes, there's a cluster where on average people are half a standard deviation down, and there's a cluster where on average people are half a standard deviation up. And we see the same for many other of these immune compartments. So for me, when you're looking at 159 samples and you see this to me underlines exactly why we do the work we do, because if you look at the cohort level, you really don't see the whole picture, right? The real picture here is that there are patients that have significant, alterations in one direction in their immune systems and patients who have alterations in the other direction. And this result at this stage simply underlines the fact that we need to look at those subsets in their own right and try to understand them for their true complexity. These differences also were seen across both disease labels, which is really interesting, right? So there were a pretty even split in the number of samples from each cluster across the different disease labels. So again, it suggests that the diversity of immune states was seen in both long COVID and ME/CFS. So the major next focus of our research is to cluster within systems to identify distinct biological groups. So, right now I'm showing you a kind of a real insight into research in progress. We're looking, we see a cohort level result. We think, okay, can we stratify these patients? Do these patients have a distribution of manifestations? If we look by systems, we'll be going much deeper into that work. When we look into different systems from our RNA data, do we see distinct biological subgroups emerge? And I think that's, for me and for all of us, the most exciting direction of our work is to try to identify those distinct biological groups and ultimately to see, do those biological groups associate with different symptom profiles, different disease manifestations, and most importantly, different clinical outcomes. So that's the RNA cohort snapshot that I wanted to share, or research in progress I wanted to share today. And really appreciate everyone joining. For all of our patients who are contributed their data, firstly, thank you so much. As I have said many times, we are honored for every patient who supports our work, who chooses to buy our test and contribute their data. I hope that today, you can see, just how promising the work that you're all supporting is looking. For our existing patients, we have some really cool new things coming soon. So I mentioned that we're doing this pathway analysis, and we will be preparing and presenting research from this pathway analysis. But this is a map of, so we're actually looking at my data. So this is my RNA sequencing data, and it's been mapped onto the pathway map of the reactome pathways. So this, in our platform, you're able to kind of zoom in and see within each system, DNA repair, metabolism, whatever it is, which of your pathways are up or down compared to the reference ranges. And then also for the pathway analysis, you can see an aggregate of which systems have more or less pathways up or down compared to the reference ranges. So this will be coming out, I think, in the next few weeks, and really excited to share this from my perspective and from the data science perspective. For all of our existing patients, I think this really is the most interesting way to look into the data, because RNA data on an individual gene basis does have variability, and the best way to look into this data is to aggregate findings into systems, and this pathway analysis does that, and it allows you to see which systems are in a coordinated way moving in different directions. And so, yeah, so for everybody who joined today, thank you so much for joining. And, for anyone who wants to join and support our research, if you, the way we operate is we try to create the most interesting tests that can provide the best information-only insights into biology. And if you do buy our tests, then and if you consent, you don't have to consent, but if you consent to research, then you contribute your data to our expanding data set. And I hope you can see today that we are really making some great headway. And at 159 samples in the RNA cohort, I think we can see now the proof of concept of our model and the value that we can create. But also, it really just underlines how exciting it is when we reach 500 samples or 1,000 samples. We may then really be able to understand underlying biological groups in a way that no one has so far. So thank you so much, everybody, for listening, and I really look forward to any questions and discussion.
Thanks, Nick. That was great. So we have quite a few questions in the Q&A, but I'm going to open it to some of our panelists first, if anybody wants to ask any questions here. Okay, I guess I'll start going with the Q&A then, but if anybody wants to, just go ahead and raise your hand. Okay, so Liam asks, for the treatment responses, was there only an improved or worsened option? How about no response?
Great question. There was no response as well. So that great question. Maybe next time I present, I'll include that in the figure as well. Yeah.
And then I'm curious. So it was just improved or worsened. There wasn't like a magnitude of improvement.
So I think currently there are magnitudes.
Okay.
So, but this for simplicity, I just plotted improved or worsened. Yeah.
Okay, great. Okay, Troy says, have you compared the treatment response differences between the two clusters? Maybe one cluster responds well to a specific treatment.
Not yet, Troy, and that is, that's exactly where our work is going, right? So, those clusters by those immune systems, those are very preliminary. I'm pretty optimistic that in weeks from now and in the next months, we'll find, much stronger separation when we look at specific systems. We'll really try to home in on which systems have strongly distinct groups. My belief is that once we identify those groups, I predict that they will, that some groups, whether we can access them or not, and how early we can access them, I don't know. But I believe that when we find separation, that will map to treatment response, because I believe that there must be underlying biological differences. So those initial groups that I just presented, I haven't yet compared treatment responses across them, but I will be doing that very shortly. So great question.
Yeah, that's going to be super interesting.
Mm.
Do you plan on testing against disease controls or sedentary controls?
Okay, interesting question. So sedentary controls, I think, is really interesting. And yeah, so basically, yes. I mean, everything is, as always, in science, you want to test everything, right? We want all data. And unfortunately, I think one of the main jobs of a scientist, which is very difficult and is a big part of the art of science, is trying to cut down to what you actually can measure and deciding what you measure. So at the stage we're at right now, we are simply matching to healthy age and sex matched controls. The reason we do that is because for us as a company to prove, to have signals that can be, potentially developed into diagnostics, regulators look at, healthy controls versus disease, right? So we have to fit into that basic system. Spiritually, however, we have a very different approach to that. So what we actually believe is that, firstly, like, I believe that everyone is on a spectrum of chronic disease, and I know that can be controversial to some people, but I really do believe that, because aging is a chronic disease. Eventually everybody has tinnitus and memory problems and cognitive slowing. So we are on a spectrum of degeneration and chronic symptom development. So for me, in the future, I really believe that comparisons within the disease are much more interesting. Certainly, I can just say openly, they're much more interesting to me than comparisons between disease and control. Now, everything I presented today was really, except the clustering at the end, was mostly comparisons with the control. But as our data set grows, I think comparisons within the disease become really interesting. For example, comparing people with one treatment response versus another, or with one symptom versus another, or with one state of a system in our RNA sequencing data. So, that kind of does map to that, right? Those are disease controls and they are sedentary controls. They're just within the same disease. To really answer your question specifically, I would absolutely love to. I think that that's, unfortunately, as always, it comes down to budget and, where do we prioritize things? But, maybe, it's actually something that I think, because we've been prioritizing, building up a healthy control cohort first, maybe your question makes me think maybe, once we get that healthy control size to something that we think is maximum that we need, maybe we should then move and start to build up some sedentary and disease controls. Great question.
Yeah, I'm going up towards the top here, kind of moving around. It's different questions. So there might be some questions from early on in your talk, but let's see. Regarding the AUC, which features from the RNA biomarkers emerged as most predictive and differentiated patient and controls?
So can you read it one more time?
Yeah, regarding the AUC, which
Yep.
features from the RNA biomarkers emerged as most predictive in differentiating patients and controls?
Great question. So that's actually not something that I've looked into at this stage. So right now, my focus on the AUC work is to, it's quite a lot of iteration, right? So it's like we're looking at, finding different signatures that separate. I haven't been into the biology deeply of them. So at this stage, it's just the priority is just separation. Once we, reach a point where we're kind of quite satisfied, then I think we will look more deeply into that biology and we'll share it publicly. I think it's really, it will be beautiful if the signatures map to biology that makes a lot of sense, and we will see what that looks like.
If B cell gene expression is down, is that just RNA expression in blood? And could it be because B cells have migrated into other tissue?
Great question. I love that question. I mean, now you will really get me onto proper nerd topics that I love. So I think the true answer to that is yes and no. Firstly, yes, it's a really great question. It's very correct, right? So B cells may mature into plasma cells and infiltrate into the tissues and be locked in the tissues. Now, the signatures in the blood, so you could say, okay, well, the B cells are maturing and going into the tissues, and so we're collecting RNA from the blood. And the question is, are we only seeing, and remember, cohort level decrease, but of course, the clustering suggests that actually there's a complete spectrum. There are some people up and some people down. So the clustering suggests, there are both extremes, right? But let's just focus on the cohort level for the sake of the question, because I think it's a really interesting one. So the yes, it could be that the B cells are maturing out into tissues, and so you're not capturing the RNA transcripts from those B cells. However, likely you would see signatures that correlate with that maturation process, right? So we know that plasma cells can be very long lasting, and so I think there would be an interesting question here of what do these transcripts do in and around crashes or baseline changes. That could be really interesting. Is there a new repopulation of B cells that infiltrate tissues? I'm not saying that's the cause of PM. I'm just speculating on potential mechanisms. But equally, when you have, for example, mature B cells in tissues, you probably will see blood RNA correlates of that, right? So you probably will see signatures of B cell transcripts that are either spilling back into the blood, or mature B cells in the tissues are signaling back to the blood, and you'll see correlates. In complete fairness, it's not implausible that you could have mature B cells in the tissues producing antibodies, and they're signaling back that there are more than enough B cells in the tissues, and so B cell production goes down, and that would fit with that cohort data. I would say that that's a very TBD thing. Like exactly which signatures map to which real state is an open question where I don't think you can say one or the other definitively. And I'd just bring back to that clustering as well, that we're seeing this spread where we could be seeing every answer to that question together averaged.
So I saw a question from Catherine Glass, and I gave you the opportunity to speak because I think that you might have some good back and forth here. So if you want to speak, you can, or I'll read your question.
Hi, yeah, I can go ahead and speak. Sorry, I'm not prepared to have my video on.
Yeah, that's okay.
Yeah, so thank you so much for your talk. I'm actually doing pretty similar work at Cornell with multiomics, primarily focused on proteomics and metabolomics. But as you mentioned, we're very limited with small sample sizes in our cohorts, and it's very difficult to get external validation cohorts or to be able to add more samples. So I really like what you're doing with this model. But so some of my questions are, what are you doing to define what sample size you need to be able to be really confident in these insights that you're sharing with patients who buy the tests? So that's my first question.
Sure. So, I mean, I think that what we do is we just try to um, build confidence as we progress, right? So in our like our kind of goal for the patient insight platform is that we populate onto that confidence ratings based on how reproducible those results are across batches within our cohort. So that's how we do that at the highest level. So we basically, we across different genes, different pathways, we look at how stable they are between bridge samples, between anchor samples, between batches, and then we populate that as a confidence rating. So that's like, it's something that we want to build as a kind of, you know, constantly improving real-time label uh, based on how stable the things that we see are, if that answers your question. And your work sounds really cool, by the way.
Yeah, no, that does answer it. So that's good that patients are already able to get that confidence rating, and it should, as you get more people buying in, then it should increase the confidence for sure. I'm
Exactly.
also wondering, what are you doing to grow the control cohort? Because controls, probably wouldn't be buying the test.
Yeah, exactly. So we fund control samples with part of the test fee, right? So the aim is that we include a 4-to-1 ratio, so one control for every 4 patients, and that's built into the cost of our test. So we run those in each batch, and that expands as the data set expands.
Okay, nice. Yeah, that sounds awesome. Yeah, so thanks again for sharing all the details of your results.
Thank you so much. And you mentioned the restrictions on data size, and I think that is, we have obviously, as I mentioned, limitations of our work, but I think that's the major advantage we have, is being able to grow in an open-ended way. And if you want to, look to validate any of your results in a larger cohort, if we are a larger cohort, feel free to get in touch anytime.
Okay, yeah, that sounds good. I mean, I think in an ideal world, patients wouldn't have to spend so much money to be able to contribute to this kind of research, and it would actually be, government funded already and everything. But I really appreciate what you guys are doing, considering that's not the world that we're living in right now, and that it hasn't been done. It's still not being done. So yeah, this is exciting. Thank you.
Absolutely. Cheers to that.
Thank you so much. Agree.
Yeah. I was going to say that's sort of, been our philosophy at Renegade Research too, that it's not happening, so we're going to make it happen. We're going to do what we need to do. So
Yeah.
patients want to contribute, we're working as a community.
Yeah.
But ideal world, yeah, wish we had more
And
funding.
I want to say on that note, like, because a lot of the most valuable testing is expensive to run, just at a baseline. There is no way to make it cheaper because it's expensive to run. So it is so incredibly valuable when those who are able to access that type of testing are so generous with their data and share that result out publicly. That makes it just so much more useful to absolutely everybody. So I want to thank those people who have been so generous with their data.
Yeah, I couldn't thank those people more, obviously. And I think, it's, Catherine, it's a really, it's a topic, of course, that as a company we have to, kind of grapple with and be careful with. But we started, with exactly the same sentiment that Tess expressed. I think we originally were from a network of patients, and we were just looking around and seeing no, significant industry mobilization. And we all said among us, we would happily ourselves pay to advance the cost of research. And so the hope is that, those who can afford to pay, are able to advance the research for everybody else. And then in the end, it benefits everybody else. And I think we kind of thought, if only 1% or 0.1% of patients could afford to pay, we'd still have millions of people who could, build the biggest data set in the world for this research. And then, looking forward, eventually, once we reach, when our data set gets large enough and when we identify signatures that have enough clinical utility, that's where we will, seek funding to develop those as validated diagnostics that then have insurance coverage and national healthcare adoption. So that's the kind of full circle. And I think some people put it nicely that, hopefully, not everyone's in the same boat, but all the boats can rise together if those who can help. So yeah, I appreciate it.
I thought Ellie came up. Did you come up to talk about the chat, or do you have a question?
Oh, just to remind people that they can still type their questions in, and we're watching them, and just appreciate Nick and everybody for being here. This is so exciting.
Thanks so much, Ellie. Yeah, we've been so excited to share these things today.
Yeah, so like Ellie mentioned, you can still put your questions in the Q&A. I'll still keep an eye on those. We'll start asking more of them. But also separately, there's the chat where we put some links and stuff that you can see. And then periodically I allow somebody to talk. So if you see all of a sudden it pops up and it's like, I'm allowing you to talk, you don't have to, but just know that that means that I might call on you. It's coming up pretty soon here. So the next one was Bert. I gave you the option to talk. Would you like to?
Yeah, I'm here.
Yeah.
Thank you very much. And thanks, Nick. This is an awesome presentation and very exciting. My question is kind of around like what you've called sort of the main goal of all this, of like predicting treatment response. I guess like the self-reported like patient surveys are good, but obviously subject to a lot of bias. Do you have kind of plans to either work with researchers or like make this data available to other researchers to potentially call on, if you get to 500 or 1,000 samples, like could that be a clinical trial population that then you could say, okay, we're looking for patients with this specific RNA expression to be in a randomized controlled trial for this drug, and using those RNA panels as like either inclusion criteria for like the patient population in the trial, or as outcome measures, like to see if certain RNA expression move with a treatment?
Absolutely. I mean, but great question, and 100%. So the way, like the pathway that we hope to walk is we will this so hopefully by early next year we will have achieved. We're going to make a US clear cap submission, right? So we're going to try and get full US clear cap regulatory approval. At that point, we can then directly use our data as clinical trial inclusion criteria. So the ultimate, our goal, we aim very high, right? The goal here is to impact care, for millions of patients. And so the path we see to doing that is to do what other companies have done, for example, in oncology. So Tempest AI is like a big, has always been a major influence to me and to us for what they did in oncology, where they basically used RNA sequencing. They spun up multi-center clinical trials, across many locations. I think they were onboarding a new clinical trial location every 12 days in the US. This is the type of Operation Warp Speed that we need, right, for these diseases. And so we plan to try to basically walk that same path, where we once we achieve the regulatory position to be incorporated into a live clinical trial, which is quite a high bar, right? Then we would try to do exactly that. So to basically spin up clinical trials around drug repurposing based on subgroups that we identify and stratify with our RNA sequencing data, and close the loop like that. So yeah, I mean, you exactly appreciate the way this can and I hope it should and will be done.
That's awesome. That's incredible to hear. Yeah, super excited. Thank you for doing the work.
Thank you, Bert. Yeah, fingers crossed.
All right. I had mentioned the chat, and then I saw a few people came up here and said that it was disabled. So I think it's set up right now. Can you see what we're putting in, but you just can't reply? If somebody can. I guess you can't reply. So if you can answer in the Q&A and tell me if you can see the chat and the links that we're sending. No, we just see the Twitter links at the start. Okay, Ellie, maybe you can see if there's something going on.
I just put 2 new links. One of Nick and Jack's previous webinar with us in 2025, and also the link to get the test and the discount code.
Okay, so I do
Hopefully
see people
are seeing that.
I do see people are starting to say that they can see it now. Those show. Okay.
Yeah, you can't reply in the chat unless we have sent you a note. It's not an open chat, so just write to us in the Q&A if you need to reach us.
Okay, now I got lost with what question I was going to ask.
That happens.
Yeah. Well, someone said not a question, just a major thank you to Nick and all the team working on this.
Yeah. Thank you so much. Really appreciate that. Thank you for joining.
How can a healthy person become a part of the control group?
Love it. Thank you for asking. We need healthy people to volunteer. So if you want to join the healthy group, I mean, I think maybe we might not have a kind of really settled system for this. I'll definitely check with our team, but please drop an email to our support email. Just if you go to the website and contact via the website and say you'd like to be a control, we'll handle it from there and we'll put you down to be a control. That's absolutely wonderful for everyone who wants to contribute. Thank you.
What is the general email address to reach the company?
Yeah, support@amaticahealth.com is the general email, and you should find that on the website, and I can drop it in the chat. I haven't put anything in the chat, but I will just, okay, I posted it right there.
Great. Oh, that just went to hosted panelists. So Ellie, could you share that to everybody?
I got it.
Okay.
Thanks. Nice.
Okay, another good question. I think I know the answer to this. Do you plan on offering metabolomics or other testing that you could pair with RNA-seq?
Yes, it's a great question. So we do. So we want to again, it's always a difficult question of, we could offer everything, right? So we could, take some time for us to get things in and to build a, standardization and to build a process and to, decide where we're going to put that time. We'd love to collect as many sample types as people want to have analyzed. We have to think where to prioritize. So I think that most likely what we will add is, I think RNA sequencing will always be the kind of central test from now going forward, because it is just so powerful. It's the richest single data set you can get. It has so much power. There's so much power for stratification via that type of data. So, but in addition to that, I think most likely we'll add proteomics alongside. So, maybe we'll expand our 31 marker test to be like a wider proteomics in the future. Then we're planning, so we're actually in the background and kind of ready to move on this fairly soon. We've been working with CDI, who make the HuProt human protein array, which is 21,000 human proteins. So it's a microarray with 21,000 wells, and it has 21,000 human proteins in in different wells. And you can take that array and you can put human serum on, and then you put human serum on, any antibodies that you have against your own human proteins will stick to those proteins in the microarray. So then you wash it and then you label, the IgG tail or one of the antibody tails, and it gives you total resolution in the body of which structures autoimmunity is focusing on. So that's called the HuProt autoantibody array. And that's something I think we will definitely add. So I think, maybe a likely lineup is we'll have RNA sequencing, proteomics, autoantibody omics, and then metabolomics. Other things we'll consider. The difficult decision is just, what do we think contains the the most valuable data, and those are open questions. So, let us know what you think in any forum, because we don't know the perfect answers, and we'll be thinking as we move.
Great. From Jay, for those of us who are patients and have friends and family who want to support, should they donate to Renegade, order Amatica kits as controls, other ideas?
Yeah, I mean, you can answer that, Tess.
Well, I'll give a plug for the project that we're doing in 2 weeks. So we're going to be, some of our team members are going to be purposely inducing PEM and making a bunch of measurements before, during, and after. And we've gotten some private donations from people, and part of that will be to fund Amatica testing for what we're doing. And we still need more funding for that to be able to analyze all of the samples that we'll be collecting. We currently have funding to analyze 8, but we want to collect 20 samples. So we actually have a time series for each person. So if you donate to us for that project, you can specifically say it's for that project. And we are a 501c3, so you can get a tax deduction if you're in the US. But I know Amatica also has other ways that you can help. So now I'll pass it to you.
Including ordering kits as a healthy control.
Yes.
If you're funding it, that's great. That's, one more test that's in that we don't need to fundraise for, that nobody needs to fundraise for. That's fantastic. On the having a time point, having multiple time points for these samples, and I will be one of those, so I should, acknowledge that. I had a chance to look at the in and out of PEM samples of one of those generous people that shared their data, and I was absolutely blown away. I could see a lot of the processes that we, a lot of us have been talking about, a lot of researchers. And it's not one model is better than the other. I think we're going to see that almost every single one of these models is active. The question is that individual variation. For this individual, I noticed a sort of pattern. I was like, well, here's this pattern. I wonder. And then they were, again, very generous and shared their DNA data for comparison. And sure enough, there was a clear driver of the slight difference in pattern that I saw in that in and out of PEM. One sample, it would have been interesting to look at, but I just don't think it would have gotten that sort of data. So being able to have that longitudinal depth is a game changer, really.
Absolutely. And yeah, I mean, I think these are, all of Renegade Research work is incredibly valuable, and I'm a huge fan of what they do. So for sure, support them. And, as we both just said, I mean, everybody who buys our test, the kind of handshake that we want to make is that we want to give you the most interesting, no one has ever created a digital platform to explore RNA sequencing data like what we've created. And so, we hope that everybody who does, buy our test, gets a really great product from it. Another really beautiful feature of the way we work is, if you get your data, we continue to analyze over time, and we build new bioinformatics pipelines and analysis architecture, and we sequentially release that on the platform as we develop it. So from a single sample, you will get ongoing continued analysis, new features. So for example, now pathway analysis will come soon, cell type deconvolution will come soon, and then you also get insights as the cohort expands. So eventually, when we're stratifying dozens of subgroups, people will be able to see from a sample they gave a year ago or 18 months ago, where they fit into a data set of hopefully 500 or 1,000. The project, which is the one exactly as Katrin just said, is the, I think we're building up PEM research. This is in collaboration with Renegade Research, and anybody who really wants to support this work, people who want to do a sample in and out of PEM, please contact us. And that is, I think, some of the most exciting data we're collecting, that we already, I think, have, between a cohort from Renegade Research, which has been supported by a donation, and our own patient community. I think we're probably already getting on for passing 10 people in and out of PEM. And so, we'll have a sample from their baseline and a sample when they're in PEM. And, that I think is going to be the most fascinating data we've collected by far. Will we see systems flipping? Will we see pathways changing in activity between those PEM states? And, at what scale will we start to see insights, I'm pretty optimistic that even at this small scale, with a longitudinal sample in and out, we should see something. But yeah, anybody who wants to support that by buying a test in and out of PM will be contributing to some really cool research. So thank you.
For that, could you actually talk a little bit about the collection method? Because I think that's very important. Normally, under normal circumstances, we would not advocate trying to induce PEM on purpose, right? This is a very, very small subset of people who have chosen to take this risk so that we all have the data. In general, not. But the collection method makes it possible.
Exactly. So, Katrina, thank you so much, because that's super important. So, but I must say, nobody should trigger PEM unless they feel comfortable to do so, right? Because personally, I'm not someone who probably could trigger PEM, because mine is a real hair trigger and can be, there's a very fine boundary where it can be very, very severe. Maybe if I was a little bit more stable, I might be able to, but please be careful of making that decision. But the way people are doing that, so I mean, because Amatica's done so much work to solve at-home collection, which is why we have 63% of patients who are house or bed bound. That makes it possible in a way that it just hasn't been. You can imagine, like, no one could try to go to a doctor's surgery after triggering PM. That's another dimension of risk. So, being able to do it at home, where you can rest, in bed for a few days or whatever it needs to be, makes that possible. And I find it very humbling that people are doing this for the greater community. I always find that a really a moving thing. But yeah, at-home blood draw means you can have it without any stress on the body. We have, and then just for perspective, so Renegade Research's group, they're doing it together in a single collection, and they're going to be triggering it together in a controlled way. We also have patients who haven't triggered it on purpose at all. So we have patients who just had 2 kits sent to them, collected a baseline tube and sent it to us, and it was in the freezer. And then they waited, and then when they triggered PEM, they then did a blood draw in the next 1 to 3 days, and then that was sent to the freezer. So and I'd really advocate for that as well. If that's a great option for anyone who can organize a home blood draw quickly, and we can support that. But yeah, just to give you guys a full idea.
Yeah, agree. The home collection is key. It's so hard to study PEM otherwise. Like, you mentioned if you're in PEM, it'd be hard to actually get to somewhere to get the samples. But then also, if you're trying to get your baseline non-PEM sample, going into the lab could give you PEM. So it's like,
absolutely
complicated.
For sure.
I gave Courtney the option to come up and speak.
Hi, thank you. I think the in and out of PM is a really great design, and being able to have that within patient comparison is super interesting. Oh, by the way, my name is Courtney Gensimer. I'm a scientist at Chronicle Bio, so I'm working in the same space. I may have missed it because I was a little in and out. But I'd love to kind of hear how you're thinking about severity and disease duration, like in the definition of subgroups. And if longitudinal plans, sort of not just like the in and out of PM, but just like longitudinal collection is like part of that approach at all.
For sure. Thank you so much for joining and for your question. So yeah, for sure. I mean, I think longitudinal is critical, right? And so I think obviously our model is driven by what patients want and clinicians, right? Clinical networks are a critical part of what we do and something we're building. But the question is, what percentage of patients will be interested in longitudinal samples? And as that grows, we're going to develop analyses specifically for longitudinal samples. But right now I think we have kind of 10 to 15% of people who are wanting to do longitudinal samples, and I think that's great. That's already very encouraging. Maybe as we roll out more utility around that, and as we develop, that uptake will be larger. What we think is that with those, with the longitudinal samples that we have, we'll be able to validate the signatures that we see in the larger cohort, right? So that has a benefit to the wider cohort overall. And then in terms of severity, so in terms of severity, I think so. We want as much resolution as possible, right? We want as much resolution on everything. Of course, you want all information. We have to kind of try to find a balance of what is, what has high compliance, right? What will have high compliance from patients, and gives us the richness of data that we want. So we collect severity according to across like many different axes. So we collect, overall functional status, different measures of function, so steps per day, talking tolerance, etc. And then we collect severity of every symptom, because I think, we, as much as the surface level, as much as the initial research needs, let's say global severity to discriminate disease groups within and potentially to track treatment responses, etc. I believe, and my biggest interest is actually in severity around specific symptoms. I think ME/CFS and all of this cluster of diseases is defined by extreme instability and heterogeneity, right? So, like I could trigger PEM and my ability to move could become significantly worse, but my mind might be completely fine. And by the way, like, you can all judge me for yourselves, but I don't have many neurological symptoms. I'm very lucky. I don't really have brain fog. I don't really have bad memory problems, but I do have really extreme physical PM. So, I think that resolution is something we see is really important, is not just to say, okay, what severity is this patient, but to say, what is the severity of this symptom and when, and how does that change? And, we want to collect as much of that as possible. So we collect severity and frequency of each symptom domain and cluster. And then I think the bigger question, which maybe we haven't got an answer to yet, and is something that I think is a huge challenge for the industry as a whole, is to handle this instability and temporal variability. How do we handle that in a research setting? It's a really complex thing. I hope I answered your question. Please let me know if there was anything else you were interested in.
No, you did. That was great. And I agree, like severity is such a subjective terminology, and how we capture that, and severity versus frequency is really hard to define in this space. So thank you. I'm excited to see where it goes.
Thank you so much. Appreciate it.
Could you actually talk a little bit about how you capture that data? Because I know you put a lot of work into making the survey much more accessible than that kind of depth of data collection usually is.
Yeah, sure. So sorry, can you repeat the first part of your question again?
Yeah, so you collect a lot of associated data. So not the lab data, but like the symptom data, the background, the history, all of that, which of course that takes a lot of time and energy on the patient's part. But I know
Exactly.
you did a lot of work on making that collection also easier, just like with the home draw for blood, kind of the equivalent for the surveys.
That's right. So yeah, we want to make things as accessible as possible. So for example, with surveys, we have a full-time full stack software engineer who's one of our really talented co-founding team. And so we've tried to create the most accessible software questionnaire possible. We're on a journey to that goal. I'm not saying how close we are to that. And I want, constant feedback and ideas on this. But for example, today, it saves every keystroke, so you can have it up in front of you and you can be halfway through a question and you can just throw it down, and you should do that if you feel you can't continue. So, there is an inherent bias in questionnaires in this area, as we all know, because some people are too severe to complete a questionnaire, and maybe that's a single question we should have is, are you too severe to complete a questionnaire as its own severity category? But we can also look at, metadata on that, of how many questions did they answer and the time it took to do so. We collect all of those things. So we want to make them as accessible as possible. They can be picked up and left anytime. We start as, it's a kind of hierarchical thing where you start high and it comes down in granularity. Our software engineers are also working on an agentic system to populate the relevant questions depending on what your answers are. We're also working on a natural language version. So some people, might want to just talk for 10 minutes into their phone and describe their experience. And that might then, we might get unstructured data there that actually gives us really important things. What does the patient think to talk about first, right? What is the first symptom patient names? What is the first medical history that patient says? That carries in it like really interesting data, which isn't captured by your typical structure questionnaire. So that's something that's in development as well. We then want to we want, for our patients, like I said, we want to create the most interesting, results and digital platform that you've ever had, to explore things. We want to integrate data over time and all of these things. And I think that we want to use the questionnaires as a way to keep people, we want to like give people a reason to want to contribute their data, with, as long as that's within people's capacity. So for example, if someone goes in and adds a new depth of data in a specific area, eventually we'd like that to immediately allow that to be to be analyzed against the rest of the cohort to give them new insights. So, the more they complete, the more they can see their position in the data. But right now, as things are, compliance is very high, and thank you so much to everyone who's contributing that data.
Okay, gave Liam the chance to come up and talk.
Yes, Liam Rosen. Couple questions. I'll make them quick. So first one, I'm super stoked about the idea that Amatica could actually be a biomarker after seeing what you presented. My question's about that. So thank you for sort of illuminating like what the sensitivity and specificity might need to be for this to be clinically validated. Is that a, hey, we don't have enough sample size yet? Or is it a, hey, we haven't done enough fine tuning? Or maybe like the third thing is maybe we'll actually not ever get there because like RNA can't do it.
Yeah, no, great question, Liam. So I think, obviously I don't know the answer today, firstly, but I think that there is certainly the big question around, if with more data, what happens, and with more tuning, what happens. Now, a lever that a lot of people pull. So globally, if you look at the research around, ME/CFS diagnostics, one of the main levers that a lot of people pull is they tighten the definitions. I'm often like maybe a little bit kind of critical of that. I often, first thing I look at in studies is, what was their inclusion criteria and their definitions, because people often define something much more narrow and then separate it much more easily, right? So, that's certainly something that can be done. So for example, you could look at, okay, what happens if we look at the AUC only for people with POTS, right? Or only for the people with some major feature of ME/CFS or long COVID? That's an open question. In terms of like, in terms of an overall diagnostic, what it would need to reach, let's say, that viability as a scalable clinical diagnostic. I think that I am optimistic that it can be reached from RNA sequencing data. There are other levers we can pull as well, like for example, generating a new feature space, right? So, different types of analyses that we can do on the RNA data that creates a new feature space, second order, either decreasing dimensions or creating new dimensions. But there's a kind of like commercial viability of this question as well, right? Is like, what becomes a scalable diagnostic which can be used all around the world in clinics, right? So ideally, for a scalable diagnostic, you want a small number of genes. If it's going to be genes, and qPCR diagnostics are new and they can absolutely work, but you want ideally a small number of genes, like 20 or less, and you want really stable performance. I am cautiously optimistic that that can be done. I didn't show this. I didn't want to go too much detail and lose, everybody with the AUC work. But one of the other threads that I didn't show is that instead of optimizing for the highest performance, we've also optimized for the smallest panel. So for example, I think we have a panel of 30 genes that has still an AUC over 0.85. Can that reach commercial viability? Let's see. Another domain which is potentially interesting is we have our questionnaire data, right? So if we layer on the questionnaire data as a completely unsupervised part of the signature, does that give us higher discriminatory power, right? So there are lots of avenues that I have to continue to go down, and let's see as the data grows. It's a great question. We don't know the answer today. The final thing I want to say, though, is that I am most interested to see what happens when we start to identify biological subgroups, because, let's say if we look at mitochondrial genes and we see there is like a really, really strong separation of 5 mitochondrial phenotypes or 2. And, if we then home in on those mitochondrial systems specifically, are we able to separate subsets, a mitochondrial state 1 versus control, mitochondrial state 2 versus control? Do we see cleaner, higher separation power there? And the thing that data scale does do with RNA sequencing is the data scale gives you better stability of signal, right? So, although like the kind of, just throwing every gene into the algorithm, we don't necessarily know that increasing data scale will increase performance, and it can sometimes go the other way. But I think, when you have 1,000, 2,000, 10,000 samples, suddenly the mitochondrial signatures can become really stable, right? And you can see a really clear biological group when you have that data scale, and that could then lead back to designing a small qPCR panel that is able to have really high discriminatory power. And let's see. Thank you so much for your question.
Amazing info. Okay, second question, follow on to that. So you talk about growing the cohort. I totally agree with the way you guys have done it, which is, bootstrapped, just getting people to pay for the test themselves, which I have done. But obviously, I'm sure you've seen the comments from some patients that can't afford it, but would love to be part of this cohort.
Yeah.
Is raising funding something you guys would consider if it's needed to scale?
Absolutely. So Liam, yeah, great question. We have these conversations all the time internally. So we are currently working on closing funding, which will allow us to expand our operation and accelerate a lot of what we do. And that would also have a significant budget for clinical validation. Sorry, not clinical validation, but analytical validation in the US in a ClearCap lab, right? So that's kind of one major path that we're going down. We think that assuming that all goes through and we continue assuming, we think the next raise we do, we would try and raise significantly more. And then, whether it's funding high interest groups to accelerate getting, specific treatment responses or specific phenotypes, or whether it's subsidizing so that we can, expand the accessibility significantly, we see that as a major goal. And I think that, the progress we're making now, I think, if we move through our current funding and move towards this regulatory position in the US, I think that's very possible. And what we've talked about is that it can kind of be architected, right? Because you can think, okay, we see these preliminary signals. Where do we need samples the most? And let's see. For now, we continue as we are, and we appreciate everybody supporting the mission. But yeah, in the future, we will see how that looks.
That's great news. I had no idea, and appreciate having raised funding a couple times myself. I know it's not easy to share stuff like that publicly because it's never confirmed. So I appreciate you, and everything that I've seen today just gives me so much hope, and I think everyone else as well.
Thank you so much, Liam. Yeah, really appreciate it.
The next person I gave the ability to speak is Genevieve.
Hi, so I'm a research director at INSERM in France, and I got recently interested in EM and in the research in EM. And besides, I'm connected with many people in France who suffer from long COVID and EM. And my first question will be a scientific one. You analyze genes, but most of our genome is made of repeats. And actually, repeats are usually silent, but they can be expressed, and especially in pathological state. So since you have already the sequencing data, it would be great if you could also look for repeat expression.
Absolutely. Yeah. So thank you so much for your question, Genevieve. Yeah, right now, and just to be clear, so that you know the methods, right now we're using a transcript library, right? So all of our research so far is on coding transcripts, right? And that's
Poly A plus, that's what you mean? Poly A plus RNA? Or all
yes.
transcripts?
No, sorry, as in all coding transcripts, right?
Coding transcripts.
Yeah, exactly. Protein coding, right? So
okay.
Yeah, and that's just where we are now. So non-coding is still in our raw data, right? So we're starting on we're starting on like a simple transcript coding library. Then later we'll expand to non-coding, and then of course there's a lot more information in that raw data as well. So we'll
you have the raw data. That's what I wanted to know. You have the raw data for that.
Exactly, that's right.
Okay, actually, it's a question we tackle, so maybe we could enter into a collaboration, maybe.
Awesome.
Well, we are interesting to start with in a very special type of repeats, and maybe we can discuss about that later on.
That would be great. Please get in touch anytime, yeah.
The second aspect is that I know so many people affected who would like to contribute, but we're in France. So is it possible to send samples?
Yeah, so it's possible to send samples from anywhere. We basically work with people from any location to coordinate with them. We do global international shipping. So this, one of the reasons that, when we were asked about other sample types, so one of the reasons that RNA is such a great sample type is because BD Biosciences make these packed RNA tubes. So they're basically tubes that contain a buffer which, on contact with your blood, breaks all the membranes of all the cells and deactivates RNases, enzymes that break RNA, and then stabilizes the RNA in the tubes. And they are stable at room temperature for 72 hours, and they are an incredible product. So it means that we do global logistics within that 72-hour period back to our UK biobank, and then everything is frozen at -ATC and stored until we extract and sequence.
But for example, you were mentioning for the PEM assay that people could freeze them, actually. That's what you were saying, no?
Yes, so we basically
waiting for PEM.
So yeah, there's a couple of ways you can do it. So I think the best way is to send. So the samples can go through one freeze for, right? So you can freeze in a regular -20°C freezer once and leave it in there, for a week. It will not make a big difference to the RNA. For long-term storage, you need to be in colder. So there's two ways. Either you can collect one sample out and freeze it in your freezer, and then collect the second and send them both back together, or you can send the first back and have that back in the biobank, and then collect the second and send it back when you're ready. So,
all right, thank you so much.
Thank you.
All right, that mention of collaboration kind of related to a few other questions. Somebody asked if you're collaborating with other researchers in the field, and then other people said, I have a background in biology or medicine. Can I collaborate with you?
Yeah, guys, thank you so much. We want all friends and collaborators. This has to be a group effort, and please, I think we'll be formalizing ways that this can all happen. Eventually we'll have research access to our data set as well in the future. That requires some infrastructure, but we'll get there. But yeah, please contact us from all quarters. We want all friends and collaborators. We're lucky to work with some of my, I guess, heroes in this space, to be honest. We, aside from Renegade Research, old friends in the industry, we work with Professor Alain Moreau, who will be kind of one of our scientific advisors. I think he's done some of the best molecular PEM work in the world. We also work with a collaborator of Renegade Research, Todd Davenport at the Pacific University, who is a really cool guy doing some really cool molecular PEM work as well. So those are our two current collaborations, and we're looking for more. So please do get in touch.
We see an Elaine in Canada for the experiment in a couple of weeks.
His work is
just in person.
Yeah.
So we gave Melissa Stutler the option, and then I'm wondering if someone on our team, Ellie, Kat, or Isabel, if you could take over reading questions for a little while after that, so I can take a little break.
Hi, can you hear me?
Yeah.
Okay. Hi, Nick. I'm glad you're here. Thanks so much for doing this. I received your box of tubes to collect a sample after the USDA confiscated it.
Oh, wow.
Yeah. Now I only have, I only have the tubes. There's no instructions. I don't know if they took it out of the box or what happened, but there's no instructions. I don't know what, they're neither for the collection or the shipping. So if you could either send that or post it on your website or something, that would be great. And then my second question is, what is the ETA on the COVID viral persistence test?
Yeah, thank you so much. I think that our support will be in touch, Melissa. Thank you so much. I'm sorry for the seizure at the border.
No, that's okay.
Yeah, I'm glad it's with you now. Yeah, for COVID persistence, we will be initiating the second phase of our development after we close this investment, our upcoming investment. The first phase, we basically saw about half of the samples that we analyzed had kind of threshold detection, and so we think we need maybe some pre-processing for that signal, and we'll announce when we start the next phase. It is a really high interest area for us. I'm really keen that we do have something that we're also speaking to a potential collaborator in that space who are doing quite cool work on it as well. So we'll announce something, I think, in the next few months. Thank you for asking.
That's super. Thank you.
Thanks.
Melissa, didn't you have
Okay.
another question as well? I thought.
No, there were just those two.
Okay. Kat, are you up to reading some questions?
I'm trying to find where it was
Where,
left
yeah,
off.
where we are.
Yeah, I'm
No,
seeing some
We
repeat.
kind of
I
jumped all
was,
over.
yeah, I was jumping all over the place. So, but I was trying to close out the ones that he did answer already.
I've been answering some as best I can.
Let's see. I don't recall hearing this one. Just curious, since I come from a background in open source software background, obviously Amatica is sharing a lot of data and info openly and being very collaborative, but it's still a proprietary testing platform company. How much intention is there to openly share the data or testing methods in the future?
Yeah.
So that's a question.
It's a great question. I mean, thank you for the question. So the open source topic is one that we think about a lot. I think that for our data set, eventually we will have research access, right? So we'll set up the infrastructure to allow researchers to have access. There's a couple of complexities. So the first is people's data protection, right? So this is health data, and it has very high standards required of security, and we consent for de-identified research on that data under certain criteria. So the eventually, I think that the way it can work eventually is that we have research applications for access to the data, and we're 100% for that. I don't think it's possible we can just open the data because of that healthcare data sensitivity. In terms of software, though, I mean, our software team is very pro open source, and so I think in that realm, we I think are very aligned. It's a topic actually which I sometimes, as someone who's worked in biotech, science and biotech my whole life, it's a funny one because I love the open source movement, and yet in biotech, it is like the exact opposite, right? Everyone has to, all of the data is kind of locked down, and everyone has to, build their development runways on that data. So it's a tension that I very much feel, but I think that the forum that I'm very confident about is getting to a position we can do research access, collaborations with university teams and researchers, and then hopefully we can kind of get the community input we need. Also, we will be so on that as we publish our work, we will be putting out preprints of our work, and at that point, we very much want all input and critique of what we do, and finding problems and community input as well. So yeah, we'll keep working on what that looks like in the future, and we'll announce as we progress.
Thank you. Okay, another question. Is there any more RNA analysis you've run but have not shown us today that you can talk about a little? Has any analysis failed to pan out, or is it just not being done yet? I was excited to see a bit more.
Great question. Yeah, I'm excited to share a lot more. I think next time we present publicly, what I started to show at the end of looking into the biology and starting to stratify by different systems, that is very, very interesting work. And I think our next public presentation will focus, deeply on that. As I said, I wanted the AUC diagnostic work to be a starting point because it's such an important proof of concept of what we do and of what will really drive our long-term success. I'm happy that you want to see more, and I'm sorry that we didn't show more today. To answer your question directly, yes, there is more research. Nothing has failed, if you like. There hasn't been directions where we've found, nothing. But the research is in process. So what we've really done is in the last 4 to 5 months, we've built the bioinformatics foundation to do this research. So a lot of what I haven't presented today, what I've spent, 20 times as long on than what I presented today is building the bioinformatics platform that allows us to do this research, right? So that is a lot of data architecture, data organization, quality control, testing for confounding factors, batch corrections, how to handle, all of the different contributors to variability, etc. So the majority of our work so far has gone into building confidence in a single data set that can have samples added over time. And from there, then over the last, let's say, 2 to 3 months, we've built up the AUC research and the beginning of our, broader disease research. So I haven't held back a lot that I didn't present today, but the things that I showed at the end is where the major focus is now, is looking into systems, stratifying by biological groups within those systems, and then layering on the data. So you have seen pretty much up to the envelope of where we are, and the next months will be very exciting. I look forward to sharing.
Thank you. The next question is, can you please speak a little more to the differences between the RNA and the 31 marker protein test? And also, is the 31 marker test also able to see PEM state?
Yeah, good question. So we basically our RNA is, so just in very simply, first, obviously, protein is looking at the functional molecules within your blood, the actual effector molecules that are doing jobs. RNA is looking at the copies of the code to make those effector molecules, right? So that may sound quite abstract, but your genes contain the information to make the proteins, and the RNAs, except for as a simplified example, and the RNAs are copies made to produce those proteins, and the number of copies tells you how much a gene is being used. So the protein data, a lot of people look to protein as the being more closely related to effect. So you're looking at actual effector molecules and levels, and that's why 99% of clinical blood testing is on proteins, right? So you're looking at the point of effect. RNA is looking at the kind of the like software running in the background, right, that's orchestrating the production of all of those proteins and the systems that regulate those proteins. So the RNA gives you much more detailed information, and I think my recommendation is that that's the test to go for if you're a new patient today. The protein is the looks at effector molecules, right? And we actually had some patients submit before and after PEM, and they had very striking changes that were super interesting. We saw changes in vascular tone regulators and immune regulators. So 100% we saw significant differences there. The RNA allowed us to kind of have a much wider net to look at more biology. So I think that's where like the major focus for that PEM research will be now. But absolutely, we saw significant changes in the protein data as well.
We're coming on 2 hours, so checking with your own energy, Nick. Make sure that we're not inducing PEM inadvertently here. So let us know when you need to. Yeah.
Thank you for asking. I think we can stop at the hour, and so maybe do a couple more questions. Thank you.
Okay, great. So when will RNA testing on biopsy samples become available? There is, hold on a second, I lost the part of the question. According to published studies, abnormalities associated with small fiber neuropathy are much more clearly detectable in tissue biopsies, and many patients with ME/CFS also have small fiber neuropathy.
Absolutely. So we are very keen to start to do RNA sequencing on biopsies. We have the capability to do that now, right? That very much can be done. The question is more around building the process and the way to incorporate that data in a standardized way and in a way that's comparable and consistent. So, I think it's a domain where we really want to expand there. One of our top clinical collaborators in New York is routinely gets muscle biopsies from her ME/CFS patients and gets, and has them analyzed for various other things. So we have the pieces ready. And, that I think the best signal we can get there is that patients have strong interest. It's really great that you're asking today. We've had other people ask. And, I do think it's really essential. I mean, when we start to look at a muscle biopsy, suddenly you then, going back to someone else's question, you then very much do, have RNA collected from tissue resident mast cells or plasma cells and neuromuscular junctions. So I think that would be a really fabulous addition to this data, and it will be driven by how many patients have interest. Because I think that one of the limiting factors, just to make it very clear, is like, we always have to analyze things in batches, right? And that's why these runs happen and everybody has to wait for each run. But the more samples we can include together at a time, the better comparability there is and the less variation there is. If you ran one sample at a time, there'd be huge variation and you would lose a lot of signal. So, for RNA sequencing, we really want to be running 20 minimum, but better 30, 40 samples per batch. Recently we've been running 50, 75, 100. So, we want to keep those numbers high. So for muscle biopsies, I guess we'll maybe start collecting interest, and thank you for that interest.
All right, one more question. I wonder if it would be possible to connect with some of the hospitals, some of the hospital on COVID departments to figure out funding and mass testing. This person has connections to Mass General and Harvard. Have you tried that approach?
Awesome. No, thank you so much. And yeah, please do get in touch. I mean, we clinical partnerships are what we see as the most important area of growth for us. So we are working, some great doctors in the US and Europe, and we want to work with more, for clinics to adopt us into their standard workup. That's a major accelerator for us, and we've got, some really good clinical partners for that. For wider adoption, the major gates ahead of us are to eventually reach, insurance coverage, and that then, I think will open, much wider accessibility. But yeah, in the meantime, doctors who are interested, absolutely contact us, and we'd love to collaborate. And one of the things that we say with our collaborating doctors is, that from their perspective, every patient that they have tested and treat helps all of their future patients as well, right? So we really appreciate that collaboration.
All right, wonderful. Thank you so much, Nick, for this amazing walkthrough and answering all these questions and all of this time. Can you, for closing, just let us know where else people can find you? What's the best way to get in touch? And if there's any difference between researchers, clinicians, patients trying to get in touch, what's the best path for that?
Thank you so much, Isabel. Thank you so much for joining. It's really a real pleasure to share this work, and it's always an honor that people are supporting this work and want to see us succeed. And we appreciate all friends in this space. And it's also really incredible to have so many great questions and excitement for this work. And I feel that there is a new energy in this area from industry, especially, right? Good companies doing good work in this area and moving it forward. So thank you all. Please, if you had questions that weren't answered, just send us an email. The support@amaticahealth.com email is a great place to reach us. Any questions, that go there. If they need me, they will find their way to me. So just please don't be shy if you want to collaborate. And you can find us on our website, amaticahealth.com, on Twitter, and Jack, who would normally be here and being a huge part of this seminar, is unfortunately not up to it right now. He's in a bit of a crash, so follow him on Twitter. He's a really great source of positivity and progress in this area. And yeah, if you want to help us, please, if you want to help us and you can, buy our test, and there's a discount code out, Amatica 10 right now for 10% off, and we appreciate everybody's support.
Is the discount code for either test, the 31 marker or the RNA?
It's just for RNA.
Okay, good to know. Yeah. And I understand you guys are going to be releasing something a little bit in the future, so one place to look would be the Renegade Research newsletter. Will it be in other places? Yeah, look, keep an eye out soon.
Absolutely, exactly. And guys, thank you so much for hosting.
Yeah, thanks for coming.
Yeah. Bye, everyone.
Bye.